Hello,
Dr.
people. People who like people. Oops. Wait, cancel that. Okay, looks like we got a few people here. Raphael, I have not looked at your video yet. Looking forward to it.
Excellent.
We got that in our future. 16 people out of a possible 50. I should do a screen grab here and give everyone bonus points. You think that's a good idea?
Yes.
Ooh. Cole Salazar is all about bonus points. Okay, there's a consensus. Doing a screen grab. Everybody gets three big warrior points. It's kind of capricious, but what the hell? Gotta support my peeps. That's you. Okay. There's 21 of us. Oh, okay. 1, 2, 3, 4. 1, 2, 3, 4, 5. Oh, that's 20. 21, including me. Okay. I did some bad counting there. Okay, so that's 20 times 3. It's 60 bonus points I'm giving out today. That cost me a lot of money. But you're worth it. Okay, so why don't we go ahead and get started here. I'm going to share my screen. Just there. I'm going to bring the chat up in case you want to chat a question. Oops. Oops. Not that. Not that. Hang on. Still figuring this out. Chat. And right on time, here comes the dog looking to go out. I'll be right back. You going? Oh. Okay, so our last lecture in cell biology, yay. I don't think it's that long. It's going to be a little on the short side, but that's okay. So just summing everything up for this week, right? Thursday is our 4th exam, which everyone is taking. After that exam, I'm hoping to get it graded Friday so you can look at it. If not, definitely by Monday morning. And then our final, I believe, is next Thursday. So you can decide at that point if you want to take that final. If you think your grade needs a little boost and the final will help, then you go ahead and take that final. If not, then you can take your 600-point class, and that's all you got. I've been talking to your lab instructor. He's got his grades just about ready, I think. Lay down. What else do I want to say about that? I guess that's about it. The only thing is, like I said, let me know ahead of time if you're going to take that final, because I don't want people opening the final saying, oh yeah, this is easy, I'll take it, or this is hard, I won't take it. You got to decide going in whether you're going to take it or not. Also, lab grades by Friday. Good. Who is this? Hello. That looks like, who was that? Riley. Oh, Riley Provost. Hello, Riley. Looking very patriotic today with your flag up. Nice, nice. Go America. We're number 1. Okay, so that's the final. Anything else? Oh, another thing. Please do the course opinion survey, the costs report. I didn't open it yet. It'll be open on Banner. Normally, as a department, we've been passing out the paper version for the last probably 5-10 years because no one does the online version. Typically, when we do online version, we get about 3 out of 50 people filling it out, which is not statistically useful for us. So please fill out uh, the online cause report, which I'm going to fire up today. Also, we had a departmental meeting yesterday by Zoom, and I think what we're going to do is everyone's adding a question about online classes and how you felt about this and how you think we can make it better if we end up having to do it again next fall. We're not really sure what's going to happen next fall. We're really looking for your feedback on that. So any questions about general maintenance of the class and stuff? Do we need to let you know if we aren't taking it or just if we are? Let me know one way or the other, I guess, whether you're going to take it or not. All right, because then we'll all be on the same page. So any other questions about the maintenance of the class? What's going on? What's not going on?
Did you post a course opinion survey?
Raphael, can you say that again?
Did you post a course opinion survey on the Moodle? Or not on the Moodle, on the banner? Or are you going to do one? It's a weird semester.
I meant to do that this morning and I forgot, but I will do that this afternoon. That's the course opinion survey I was just asking you to do. So I will get that loaded up this right after we finish here. So it'll be there for you.
Feels weird doing them this semester because everything's weird.
Yeah, everything's weird this semester. I'm kind of wishing you guys would have your pictures up because I want to know if your hair looks as bad as mine does at this point. But of course, you're not going to do that because you don't have to. Okay, I get it. I get it. Wait.
I am aiming to try to see if I can get a mullet.
A mullet. Oh, I had the most intense mullet back in the 80s. It was a good thing. It was a good thing. But hair today, gone tomorrow, right? Okay, so let's go ahead and get started. I wrote out my notes to go with my PowerPoint yesterday, and then I realized what I wanted to talk about first was in some of the last PowerPoints. So I'm skipping ahead to slide 60. I was going to take those two slides and put them up in the front, and then I thought, well, now I have to go through all my notes and change the slide numbers all the way down. Too lazy for that. So we're going to start on slide 60 and then back up to some later slides afterward. So in the notes, the first thing we're talking about then, I'm just bringing my notes up so I can see them. Cancer. What is the cause of cancer? Basically, cancer is caused by DNA mutations, as far as we know at this point. There are some viral cancers that we'll talk about, which are not DNA mutation related. But for the most part, they're caused because of DNA mutations. Those viral cancers, by the way, the reason why they're cancerous is one of the genes within the virus is an oncogene. So it brings its own oncogene with it, and that's why they're cancerous. But mostly they're DNA mutations. Why? Why do DNA mutations cause cancer? And that's because two different things. The first thing is it tends to convert proto-oncogenes into oncogenes. So a proto-oncogene is a gene that makes a protein that stimulates cell division. So everything we've been talking about, all these proteins are made by proto-oncogenes. So your growth factor, if we go to slide 60, growth factors, PDGF, FGF, EGF, if the cell loses control of that, then it goes from, it is a proto-oncogene, cell loses control of it, it becomes an oncogene. So for example, a cell may lose control over the production and release of EGF, releases it, and then it responds to it. So it's like autocrine stimulation of itself by using the growth factor. Receptors are things that are often mutated in cancers. So you can see there are a couple of examples. The epidermal growth factor is really big in breast cancer. It's an epidermal tissue. And what can happen, we'll show later, but what can happen is they can mutate so they no longer have their extracellular hormone binding parts, and they bind together without any hormone. So boom, constitutively active. That's a big word here, constitutively active, which means they're just constantly active. They don't have to be turned on. And that's kind of the point of this whole proto-oncogene to oncogene mutation, in that the proto-oncogene is a protein that can be controlled. The cell is controlling it, so it only divides when it needs to divide. An oncogene is then an uncontrolled proto-oncogene. So now you make a protein and it's no longer controlled, so you don't have to have the growth factor bind to activate the whole pathway. Something in the middle of the pathway is automatically on, so everything downstream happens despite the fact you're not stimulating that cell anymore. So anything along that chain is made from a proto-oncogene that can become an oncogene. So just to skim through a little bit here, your growth factors, your receptors, your G proteins, especially your small monomeric G proteins, RAS being your biggest one, kinases, RAF, RAC, MEC, all those kinases, JAK from the JAK-STAT pathway. All these things are activating transcription factors. The transcription factors themselves can be oncogenes. MYC is a really big one because MYC itself can just totally activate that cell. So MYC is a really big oncogene when it becomes mutated. FOS and Jun as well. Cell cycle regulators, cyclins, CDKs, BCL2, which is an anti-apoptotic. I think I throw them into the other category though, but they're put here as cell cycle regulators. Okay, I'll get to those later, but cyclins and CDKs anyway. All right, so that's one big category of DNA mutations that cause cells to become cancers. The other is tumor suppressors. So tumor suppressors are blocking cell division. So when you knock out a tumor suppressor, it causes or allows the cell to become cancerous. And the first one found was RB, and it was kind of like this big shock. It's like, oh no, it gets knocked. It only causes cancer when it's not there, not when it is there. So it was like, what the hell is going on? So they figured out RB then is blocking cell division, right, by binding to E2F and blocking the cell from going into the S phase. And you have to knock out both genes, right, both copies of the RB gene for the cell to become lose control, basically. So in terms of tumor suppressors, we've got two different types, the gatekeepers and the caretakers. The gatekeepers are proteins that are blocking cell division, and the caretakers are ones that are either sensing DNA damage or correcting DNA damage. Gatekeepers are things like RB, CDK inhibitors like P21, P15, P16, P27, things like that. So when you knock those out, the cell loses a certain amount of its control over cell division. Your caretakers then are things that are here we go. Caretakers are things that are fixing DNA damage. BRCA1, BRCA2, really big in breast cancer again. It's been I keep bringing up breast cancer because there's been a lot of research into it. It's a really, really bad cancer. And so they've done a lot of work on it, and they found a lot of the molecular mechanisms of it. So caretakers, P53 you can put in there, ATM, ATR, DNA repair enzymes, MDM2. They put MDM2 up in the other category, but I put it down here because it's really involved in that control of DNA damage stuff. Pro-apoptotic proteins. So your pro-apoptotic BCL2, if it's overexpressed, then the cell doesn't go into apoptosis, right? So say you've got a whole lot of DNA damage, P53 gets activated, that makes PUMA, but you've got an overexpression of BCL2. And remember with the whole BCL2 thing, the pro-apoptotics have to outnumber the anti-apoptotics. So if you're overexpressing the anti-apoptotic BCL2, the cell doesn't go into apoptosis even though it should. So pro-apoptotic BCL2s are also tumor suppressors in that respect. Okay, and here, I think there's a picture of a kid here. Yep, there he is. So this is xeroderma pigmentosum. which is a XPA here, DNA excision repair. So you know how, I don't know if you talked about this in genetics, I kind of breezed over it here. When DNA is hit by ultraviolet radiation, it activates thymines, and thymines then covalently bind to each other. That's a mistake, right? So then the excision repair means this enzyme comes in, cuts at either side of that thymine dimer, pulls it out, puts the right DNA back in. If you don't have that, then you're very sensitive to UV light and UV mutations. And you can see this guy in his, actually, he's pretty COVID compliant there, isn't he? I think he would be okay today. Okay, so that is why DNA damage is causing cancer, because it results in activating proto-oncogenes to become oncogenes, and also in knocking out tumor suppressors. Questions, comments? Any comments about that? Bringing my chat up. Do we need to let you know if we aren't taking it? I already did that one. Okay. Sydney's got a nope. Sydney's all nope today. Okay. All right. So let's, as long as there's no questions, let's talk about where DNA damage comes from. Bum, ba, ba, bum. No surprise here. Cigarette smoking. So what's this line here? Sorry. There, gone. So where did DNA damage come from? Chemical damage is one source. So you've got different environmental toxins. One is polyaromatic hydrocarbons, which is basically when you take any kind of organic material and burn it. If you don't completely burn it to CO2 and carbon dioxide and water, then you get these partial products of partial combustion, which are polyaromatic, and they are carcinogenic. So cigarettes, anything you smoke is going to have a lot of polyaromatic hydrocarbons in it. Also, when you barbecue, you get a little bit of black char on your food. That's PAH, which is also carcinogenic, but much lower level than smoking 40 cigarettes a day, much less smoke. They say back in the day when there were no chimneys, people had a hole in the ceiling of their building and had the fire right in the middle of the room with the smoke going out the hole. The whole house is filled with smoke. They probably had higher rates of cancer back then for that reason. Okay, so you've got your smoke. You've got the Hudson River, right? Everybody knows about the Hudson River. GE dumped their polychlorinated biphenyls into the river for a long time because the river got rid of it, right? You hit it in the river takes it away. It's not your problem anymore. So now it's our problem because it, got into the sand and the silt and everything, and now it's all part of the Hudson River. Do not eat the fish from the Hudson River. Those are also carcinogenic chemicals. And there's a lot of other sources. Those are just the ones I have chosen to talk about. Now, when it comes to carcinogens, there are two different types. Another thing about carcinogens here on slide 31 is that some things are not carcinogenic till they hit your liver, and your liver turns them into carcinogens. Those are called pre-carcinogens. So I talk about cytochrome P450s a number of times in this class because they're kind of interesting. And they are just kind of generalizing enzymes. They're just grabbing things and changing their structure, because the whole idea of the P450 is you don't know what toxins the environment's giving you. So anything that looks different than what you're used to, right? Not a sugar, not an amino acid. Here's a weird chemical. Let's get rid of it. So anything weird that comes in through your intestines goes to your liver. Your liver tries to change it to get rid of it. Sometimes, usually, it does a good job of getting rid of those toxins. Sometimes it makes them more toxic. So this is an example of that. I think I don't know how many of you took my drugs class, but I talk about this with THC. There's a difference in smoking marijuana versus eating it, in that when you smoke it, you're getting THC, delta 9 THC. When you eat it, it goes to your liver, and half of it gets converted into delta 11 THC, which is a little different than delta 9 THC. So again, your liver is activating a chemical instead of inactivating it. Okay, so anyway, pre-carcinogens and the liver. The liver can turn things that weren't soluble into, or weren't carcinogenic, into carcinogenics. All right, so when it comes to carcinogens, you've got two different types. You've got your initiators and your promoters. So DNVA, I'm not really sure what that stands for, but it's an initiator, which means it'll go in and create some DNA mutation that makes the cell precancerous. So it's not a cancer cell, but it's susceptible to cancer. And then this other one, croton oil, activates protein kinase C. It's a promoter. So the cell will become cancerous if it has this, but it has to have a constant supply of it. And with just the initiator alone, you don't get cancer. With just the activator, you don't, or the promoter, you don't get cancer. But you get the initiator first, and then the activator, and that's what produces the cancer. And some of the activators are things that are already in your body. So again, let's go to breast cancer, because it's something I know a little bit about. Sometimes you get breast cancers that have an initiation event that involves upregulating an estrogen receptor. Well, the estrogen receptor doesn't really do anything unless there's estrogen around. But if you're a female, you've got a lot of estrogen. And so the promoter is actually a natural chemical in your body. So you've got the mutation that causes upregulation of a receptor, and then it's promoted by the estrogen in your body that upregulates it. And that's why one of the nice drugs for that is an estrogen antagonist called tamoxifen. We always have to bring drugs into this, don't we? Oh, well. Okay, so here's what I was talking about before with DNA damage from ultraviolet light. When ultraviolet light hits DNA, if you've got two thymines together, which randomly occurs throughout the genome in different places, those thymines will then become activated, ionized, and then they bind to each other. That makes a kink in the DNA. So if you've got all your repair enzymes, not a big deal. Go in, cut it at either side of that kink, take that DNA out, put new DNA in, and fix it. But if you've lost some of those DNA repair enzymes, that's where you end up in trouble. Excuse me. Okay, so virally caused cancers. This is the next thing. Oh, so sources of ionizing radiation, the sun, right? UV rays, X-rays, gamma rays, the universe in general is hitting us with cosmic rays all the time. And also you've got nuclear decay. So this is when you've got some kind of radioactive nuclide, like a radioactive isotope of a chemical, and it decays. So you have a proton that falls apart, becomes two different particles, and one of them is a high-speed positron, and that's a beta radiation. So that can penetrate and cause DNA to be activated, just like the UV light does. So that's what the nuclear decay is all about. We don't need to know a lot about that, though. Viruses can also cause cancer two different ways. Here's HPV in slide 34. So basically, HPV has got some oncogenes as part of its genome. So it infects the cells and it causes the cells to divide. And 0.8% of them will actually go on to form cancers. Also, let's see if I have it here. I think I do. Oh, nope, too far. I've gone too far. I'll talk about it later, but retroviruses can insert themselves into the genome, and that can activate other genes that are nearby, proto-oncogenes that are nearby. I'll talk about that later when we talk about DNA mutations that lead to cancer. Okay, so how does cancer form? I'm glad you asked. Slides 35 to 39. So let's just go through these slides and then I'll talk about what they're saying. So here in slide 1, we take some mouse melanoma cells from a different mouse that has no doubt died already. Take those cells, inject them into a mouse. And then that mouse will develop some melanoma metastases, right? You're putting them into the bloodstream, they get into the lungs, and then they form new tumors in the lungs. So you've got these metastatic cells, you put them into the mouse, they form more tumors, no surprise. But you take those metastases out of mouse number 1, you dissolve them into cells, you take the cells, you put them into a different mouse. This new mouse gets even more metastases. Take those tumors out of that mouse, make them into cells, put them in another house, you get even more metastases. So the basic idea is the longer a cell gets to live, the more mutations they develop and the more aggressive they get. So early on, a cell may just be benign, but more mutations build up and it goes from benign to malignant. More mutations build up, it goes from malignant to metastatic. And as it gets more and more mutated, it will go to more aggressive metastatic tumors. So that's why it's important to catch cancer early, because cancer cells are genetically unstable. They'll keep changing and changing and changing. So you want to get them before they get too aggressive. What else did I want to say about that? Aggressive mutation, just can't remember. Okay, probably wasn't important. Okay, so here's another example of the same kind of a thing, the stepwise development of cancer. And I start out here just talking about, on slide 40, talking about how the cells in your colon are replaced. Colon cells are epithelial. You've got pretty much the external environment going through your colon, right? When you think about your body, your mouth to your anus is a tube, and what's inside the tube is really not really inside your body. Nothing's in your body until it goes through those epithelial cells and gets into your actual body. So that's really the external environment going through you, and you're extracting nutrients from it as it goes through. So this epithelial layer of cells here in your colon is the barrier between you and the outside world. And because of that, it gets hit with a lot of toxins. You know, you eat food, there's toxins in it, so it's got a high degree of toxins. And also there's some physical damage that happens to it by things scraping through. I don't know about you, I like to eat glass every now and then, and it causes a little damage, but it gets fixed. But anyway, cells get scraped off here as stuff goes through your colon. So your colon is set up to constantly replace itself. It's constantly turning over. So here, and that's done through this crypt system. You've got these crypts here, and I talked about basement membrane here. Right on the outside of the crypt is the basement membrane, or the basal lamina. It goes all the way around the crypt and across the bottom of this. So that's really the separation between the outside world and inside you, is that that basal lamina layer. So down here at the bottom of the crypt, you've got stem cells actively dividing, divide. The Wnt pathway is really important in stimulating that division. So you've got a lot of beta-catenin here, catenin, whatever, and they're dividing. Every time a cell here divides, it makes two. One goes this way and the other one stays here. Now, as this cell divides, it keeps pushing cells out this way. And so as cells are getting pushed out this way, they get pushed up that crypt. And as they go up that crypt, they get exposed to less and less Wnt. And so they get to a point where Wnt signaling just stops, and they differentiate and become surface epithelial cells. and then they end up migrating out onto the surface and they become surface cells, and eventually they'll die up there. I think apoptosis might be part of that death. Not 100% on that though. But that's the normal life cycle of a colon epithelial cell. Stem cells make them, they get pushed up and up, they differentiate, and they hit the surface. Wnt being really important. Okay, so how does colon cancer happen? If you ever heard people having the endoscopy goes up the poop chute there, looking for polyps, right? They're looking for colon polyps. This is where the polyps come from. These crypts then, if you get a mutation here, see these cells here at the bottom of the crypt are supposed to be no longer dividing but heading towards differentiation, but they stay like stem cells, so they keep dividing. And they can't keep pushing up, so they push in and they push into the center of that crypt. And here we're talking about a mutation in APC or beta-catenin, the Wnt pathway. APC, remember, was one of the 3 proteins that was holding GSK3 beta in the right orientation so that it phosphorylates beta-catenin, beta-catenin gets broken down, right? You lose APC, and now that GSK3 beta doesn't work, beta-catenin builds up. So you've got some kind of signaling problem there, and the cell keeps dividing and dividing. Now, as time goes on, you've got this small tumor down here, undetectable in the bottom of a crypt, and then you get another mutation. Another mutation happens in one of these cells. That cell gets more aggressive, it divides and divides, outcompetes all the other cells, and it becomes the dominant form. Here we've got a BRAF mutation, so a RAF signal in that signaling cascade gets constitutively activated. Now they're dividing even faster, and they fill up that crypt. Then you get another mutation. Here we're talking PI3 kinase gets mutated, so it's active, or PTEN, which turns off PI3 kinase. And then boom, now you've got an even bigger polyp, and this is when the polyps come out into the colon, and that's what you're detecting with that endoscopy. And then also, as they get more and more aggressive, more and more mutations, you can see now they've broken through that basement membrane and they've actually invaded into the body. And at that point, those cells, when they go metastatic, go into the bloodstream. All the blood from your colon goes to your liver. Now you've got liver cancer, right? And the liver is extremely important. I think you can live about 3 days without a liver. So that's how colon cancer kills you. It's not because of the colon, it's because of the liver. So anyway, point being, stepwise development of cancer. You get one mutation, cells get a little too overactive, and then some of those overactive cells will lose tumor suppressors. Now they get genetically unstable. Then you get more mutations that activate more growth. Now they're growing faster. You get more mutations that cause them to, release plasminogen activator. Now they're breaking through the basement membrane. More and more mutations cause them to be more and more aggressive. Genetically unstable, remember that. Okay, so here's our APC pathway. The normal control of it here with Wnt protein activating beta-catenin, and here you've got no APC, so it's always on even without Wnt being present. Kind of talked about that before. Okay, DNA mutations that lead to cancer. In what way can DNA be mutated that will make proto-oncogenes become active? That's another way of saying that. So one way, I don't know about you, but whenever I think about DNA mutation, I always think about point mutations. So a point mutation is when you mutate one base or a couple of bases in the gene, in the coding region of a gene, and that causes a change in the amino acids. Now remember how unlikely this is. I don't know if you had me for Bio 101, but I always say when you do a point mutation, most of the time that point mutation is going to be in non-coding region, so it really isn't going to change anything. As long as it's not in a control element, it's just in a non-coding region, an intron, a space between genes, it's not going to do anything. So most point mutations are silent. If it's inside a coding region, odds are pretty good it's just going to screw that protein up and it won't be functional. Again, no big deal. Maybe the cell will die, so what? But occasionally it will change the function of the protein and it'll become more active. And that's kind of the basis of evolution to a certain extent, but also the basis of cancer. So here, you're talking about a point mutation changing a protein. And the example I like to use is RAS. So RAS normally, as we've talked about in the class, gets activated by a GEF, a G protein exchange factor. And so it kicks out GDP, and GTP gets picked up, gets active, and then it starts, and it does its thing, and then it interacts with GTP's activating protein, and that causes it to hydrolyze GTP and get turned off. That's your G protein cycling. So there are 3 different mutations that can happen to RAS, and they're all in this active site near the GTP. And if that happens, then you can't break down GTP. So RAS gets activated once and it stays active. There's no turning it off. And that's how a point mutation can cause cancer. Okay. Gene amplification, I've kind of alluded to this several times already, but normally a protein, like a proto-oncogene, is being regulated on and off, and also level of expression. Excuse me. And DNA, during replication, crazy things happen. So a gene can just get flipped around and then duplicated. Now you've got 2. In the next round of replication, it gets replicated. Now you've got 4 copies of it, 8 copies of it. And you get big chunks of DNA that does this weird replication where it ends up with multiple copies of a gene. So when you think about something like MYC is not regulated by the cell at all, except by regulation of its transcription. So once MYC is made, it's active. You get too many copies of MYC, now you get too much MYC. So excess overexpression of the protein can cause it to be active. Also, something like a G protein, no, what am I saying? Tyrosine kinase receptor. Normally, tyrosine kinase receptor need hormone to dimerize. If you make like 10 to 100 times more tyrosine kinase receptor, they start activating themselves without any hormone. Okay, there's just so many there, they bump into each other and activate. So excess amount of protein can cause that to happen. And here's 2 different mechanisms here. Here's an activating mutation where, I said this earlier today, you cut off the cytoplasmic side of the receptor, and the receptor automatically dimerizes and activates. That's kind of like a point mutation effect. Here is overexpression. So here they actually have the hormone, so it's over responsive to the hormone, but also it could just be, just the receptor itself activates itself when there's too much of it. What do you mean by make them into cells? Can you restate that, Adelina? I'm not sure what you mean, make them into cells. What did I say?
It was a couple slides back when you were talking about the mice and metastasizing cells, and I just didn't know what you meant.
Oh, I said that pretty quick. So what I meant there was you take a tumor out, right? It's a tissue. Tumor is a tissue. You hit it with collagenase, and now it's individual cells, and you inject those individual cells. Does that make sense? Okay, good. All right, coming back forward, DNA mutations. Okay, so another DNA mutation is a translocation. Again, let's back up. Sorry. Translocation. So chromosomal translocation. Again, weird stuff happens during replication sometimes, and DNA is easy to screw up. So during replication, the DNA is all expanded, and now this strand breaks, and this strand breaks, and now you connect them, and now you've got a chunk of one protein or chromosome gets stuck on a chunk of another protein, and you're swapping chunks of protein. And you get basically new chromosomes. So this is an example here in 49 where you've got chromosome A and chromosome B. Big chunk comes off A, big chunk comes off B, and they switch. And that's called a balanced translocation. Chunk of this one, chunk of that one. And now at this junction, there's a couple different ways this can cause problems. One is at this junction where they hook together, you could be making a brand new protein. You know, if this happens in the middle of a gene, now you can take two different genes, you stick them together, and now you've got a chimeric protein. And, or, let's do the chimeric protein first. Okay, so here is CML, chronic myelogenous leukemia, where you take a chunk of chromosome 9 and a chunk of chromosome 22, and you put them together, and right at that junction, you had two different proteins. You had BCR and ABL. So BCR gets broken here, ABL gets broken, right at the beginning. So you take this chunk of BCR, you stick it onto ABL, now you got a brand new protein, and this is an unregulated kinase. So this BCR is, where was I? Here it is. Here's the kinase here. So here's the kinase. Normally in BCR, it's regulated, but when you connect it to ABL, it's no longer regulated, and you got an active kinase. So point being, what the hell is your point, Slish? So the point was, we did a chromosomal translocation. We made a brand new protein, a brand new chromosome, and at that junction between the two chromosomes, we made a new protein by taking half of this one, half of that one, and sticking it together, and it's no longer active or no longer controlled. Here's another thing that can happen with chromosomal translocation, and that's overexpression. So Burkitt's lymphoma, we've got chromosome 8 and chromosome 14. So chromosome 8 is where MYC is, and you would imagine in an adult cell that's post-mitotic, you would like totally condense the DNA where MYC is, right? Have it all tied up with histones and ultra condensed so that we're hiding MYC. We don't want it to be active. And then over here in 14, there's an extremely active region. It's the region where immunoglobulins are made. So if you're a white blood cell that makes immunoglobulins, that part of the DNA is always expanded because you're constantly using that DNA. So what happens in this Burkitt's lymphoma is you get a chromosomal translocation between 8 and 14, and you take that chunk of 8 where MYC is, and you put it next to that active region of 14, and now the DNA is expanded and MYC is accessible, MYC gets made and overmade, and boom, now you've got a cell that keeps dividing. Okay, so that's another way that chromosomal translocation can cause cancer. One, by putting genes that should be quiet into active areas and now they're overexpressed, and another by making brand new proteins, fusing two proteins together and getting a brand new one that's unregulated. There's your Philadelphia chromosome. I point this out because this CML is a disease that some of these new strategies to stop cancer are being used. In that, like the old cancer therapy was always kill dividing cells. So all these chemotherapy agents were basically toxins, poisons that will kill cells in mitosis. Things like binding to topoisomerase, binding to helicase, binding to microtubules and screwing up the microtubules. And the idea was, it's a shotgun approach, but the idea was cancer cells are dividing faster than other cells. If we kill all dividing cells, then let the person heal. The good dividing cells, like stem cells, will come back. The bad dividing cells, the cancers, will not. Kind of works some of the time, which is good when it does. This is a more specific goal, which is we know this protein is bad in CML. So we get the molecular structure of the protein, we design a drug that's going to fit into that kinase domain and block it, boom. Now we're not killing every cell in the person, we're only killing the cells that are overactive. So that's the new line of chemotherapy, which is becoming really, really popular. My internet's becoming unstable. I'm hoping I don't drop out, but if I do, blame my internet. Okay, so DNA not overexpression, local DNA rearrangement. So the last thing we talked about was big chunks of chromosomes being moved. Now we're looking at within one chromosome. Again, DNA is so friggin' weird. A big chunk of the chromosome can come out and flip around and stick back in, right? Remember, it's not linear, it's a rope. And two ends of the rope come together and it flips around, and now you've got big chunk of DNA flipped around. You can have a chunk of DNA come from here over to here. Things happen. And so why is that bad? So here is a couple of things that can happen. Here's a deleted chunk of chromosome. So that is going to change the structure of your chromosome and can activate some proto-oncogenes. And here's an inversion, slide 56, where it breaks in two places. Instead of losing that whole chromosome, it flips around and sticks back in. So you've got this reinserted piece of DNA here. And now that can make new proteins. And this is a really interesting one, which is a combination of tropomyosin and the NGF receptor. So you remember tropomyosin from way back in chapter 14. Tropomyosin is laying along the actin microfilaments, the thin filaments in skeletal muscle. So this tropomyosin then is covering the actin binding sites. This is review for the final, by the way. And then here's the troponin complex that is holding the position of that thing. And the key thing about this, which I was saying at the time, I keep giving you hints at the beginning of the class of where we're going eventually. So tropomyosin is a coiled coil. So you take tropomyosin, you put it in solution, it will spontaneously form a coiled coil. Here's the FGF receptor. Well, it's actually the NGF receptor that we're concerned with, but it's structurally similar. And the whole idea with the receptor is you get two of them together and that activates them, right? So with this mutation, you've got tropomyosin here and the NGF receptor here. The chromosome breaks in two places, flips around, sticks back in, and now you've got a whole new protein over here. Here you've just got a dead protein, but over here you've got a protein that's tropomyosin, nerve growth factor receptor. And so the coiled coil keeps those receptors coupled. So as soon as the protein's made, it's active, it's constitutively active. And so now the cell is dividing out of control. So that's how local DNA rearrangements can then cause cancer, turning proto-oncogenes into oncogenes. Retroviruses. So interesting story. When I was finishing my PhD, I wanted to go into a lab in MIT that was doing some retroviral mediated gene therapy. It sounded really exciting at the time. It's like, okay, we know cystic fibrosis is caused by this gene that is screwed up. So why don't we take the good gene, put it in a virus, infect those cells, a retrovirus, have it go right into the genome, boom, you fix the problem. Well, the problem with retroviruses, you can't really tell where those genes are going to go. The retrovirus is an RNA virus. It binds to the cell. I think the COVID is a retrovirus too. Binds to the cell, releases its RNA into the cell, and one of the genes on that retrovirus is a reverse transcriptase that turns the RNA into DNA. Then you get double-stranded DNA being made. That DNA inserts into the genome, and now it's there forever. And then it starts making its viral proteins and viral DNA or RNA, and then it makes more viruses. Well, the problem with retroviruses is this whole idea of inserting into the genome, kind of like we talked about with MYC earlier. If you've got MYC in a quiet part of the genome, and then this virus inserts next to it, now MYC is in an active area, so the cell starts dividing. So retroviruses can cause cancer, not because of the genes in them, but because they insert randomly into the DNA, and they can turn a proto-oncogene on when they do that. And that's why you don't hear anything about retroviral mediated gene therapy anymore, because it was like, this doesn't really work. What do we use to put new genes into DNA now? CRISPR-Cas9, send me a dream. CRISPR you can put it exactly where you want to put it. So you take your fixed cystic fibrosis gene and you put it directly in the middle of the bad gene, boom, you fix the problem. So that's the future now. Okay. CRISPR-Cas9, send me a dream. Okay, and now we're back to the beginning. Okay, so I guess that's all I have today.
You should show the video I posted. It's really good. It's a really interesting. It mentions a really interesting way to treat cancer. Do you want to hear what it's called?
Absolutely not, because I'm going to go through the last couple of slides and then I'm going to play it.
Okay.
Okay, so all right, so the rest of it just goes through the hallmarks of cancer, which I talked about already: self-sufficiency in growth signals, insensitivity to anti-growth signals, evasion of apoptosis, limitless replicative potential, telomerase. Oops, wrong way. Sustained angiogenesis, tissue invasion, and metastasis. All right, so let's pull up that video, because I haven't seen it yet, but I know Raphael long enough now that I know I can trust him. It's not porn, right, Raphael?
Yay, thank you.
Joking, you wouldn't do that. All right, let's watch this. And I know I have to change my settings here. CRISPR-Cas9, send me a dream. Let's get this started. Let's get this puppy going here.
Cancer is a creepy and mysterious thing.
All right, I know I have to change my share. New share. Share that. Okay, let's go.
In the process of trying to understand it, to get better at killing it, we discovered a biological paradox that remains unsolved to this day. Large animals seem to be immune to cancer, which doesn't make any sense. The bigger a being, the more cancer it should have. To understand why, we first need to take a look at the nature of cancer itself. Our cells are protein robots made out of hundreds of millions of parts. Guided only by chemical reactions, they create and dismantle structures, sustain a metabolism to gain energy, or make almost perfect copies of themselves. We call these complex chemical reactions pathways. They are biochemical networks upon networks, intertwined and stacked on top of each other. Most of them can barely be comprehended by a single human mind, and yet they function perfectly. Until they don't. With billions of trillions of reactions happening in thousands of networks over many years, the question is not if something will go wrong, but when. Tiny mistakes add up until the grandiose machinery gets corrupted. To prevent this from getting out of hand, our cells have kill switches that make them commit suicide. But these kill switches are not infallible. If they fail, a cell can turn into a cancer cell. Most of them are slain by the immune system very quickly. But this is a numbers game. Given enough time, a cell will accrue enough mistakes, slip by unnoticed, and begin making more of itself. All animals have to deal with this problem. In general, the cells of different animals are the same size. The cells of a mouse aren't smaller than yours. It just has...
Okay, bad news. Doesn't seem to be working very well. I was working fine for me on my computer, but it must be sending a video like that out over Zoom while also doing my video and your voices. It's just not too much. Looks pretty interesting, though. I'm going to look at it later. I think everyone should.
It's really good. The channel is Kurzcast. They're a German channel that does videos on science topics.
Oh, yeah.
And they do months of research before they make a video to make sure they have all the correct material they need.
Sounds cool. Okay, well, that's about all I have for today. We will have an exam in 2 days. It will be on Moodle. So, question. Someone told me like a week or two ago that you were supposed to have an organic chemistry at the same time test. Like it started in the morning and went till like, I think they said it was from 10 until 5 p.m. or something. Typical organic chemistry test. Is that
Yeah.
still true or no?
He moved it to finals week.
Oh, good. Okay. So then we'll just have our exam at 11 like normal. Okay. Hour and 15 minutes is not going to be longer than a normal exam, just a regular old exam. It'll be like the last one where there's time limits to try and keep you honest. Please, no cheating. I know it's a hard semester, but don't cheat. Cheaters never win and winners never cheat. Remember that. Okay. Any questions about the test? I'm going to write it tomorrow, so I don't have it yet. Okay. Stoke. Who's Stoke? No one's telling me. Qualm. Oh, wait a minute. You know what? Okay, who do we got here? I said I would give everyone 3 bonus points, and some people showed up late, so they will get bonus points as well. Booyah. Booyah. Okay. So I don't have anything else. Shall we call it a day?
If so, have a good day, everybody.
Everybody good? No questions? No comments? What topics are on there? Kwame, you always ask this question. Everything we did since the last exam. Everything we did since the last exam. Can you put the answers up for the practice test? Oh, Stokes, Wayne Stokes. Okay, so yeah, I will put I think it was exam from 2017. I'll go through and fix that. I will put the answers in for that exam. Probably not the easy ones. I'll just do the essay questions. I mean, the easy ones you can just look up. Any questions, comments? We're going to call it a day. Okay, I'm going to say goodbye. Miss you guys. Miss your faces. Is there a game? Yes. Thank you, Rana. There is a game. Don't forget, in Moodle, there's a game that's worth bonus points. So don't miss that. Okay, everybody good? Thumbs up. Everybody leaving. All right, goodbye, guys. Later, dude.